Skeletal muscles are made up of fascicles, which are bundles of skeletal muscle fibers or cells. Muscle cells are composed of fiber bundles which can contain hundreds of myofibrils. Myofibrils are the cylindrical structures that carry out contraction, otherwise known as sarcomeres. Myofibrils consist of bundles of myofilaments. The sarcomere is the distance between two Z lines, which are formed by the joining points of the thin filaments. Z lines provide the point of attachment of actin filaments. Another membrane, the M band or line, is the point of attachment of the myosin filaments. The dark band or A band corresponds to the length of thick filaments. The lighter I band represents areas where the thin filaments are present, while the H band is where both actin and myosin run parallel to each other. Protein filaments compose primarily of actin and myosin. The actin, troponin, and tropomyosin forms the thin filaments, and the myosin forms the thick filament.
An axon together with all the muscle cells it innervates is called a motor unit. A muscle contraction is an elaborate process starting with the axons of nerve cells from the spinal cord that are attached to each muscle fiber. The axon and muscle fiber are specifically called the neuromuscular junction. Muscle contraction begins first with an action potential passing down the nerve to the synaptic knob. The action potential stimulates calcium channels in the membrane, allowing calcium to move into the synaptic bulb through the voltage-gated channels. Increased calcium causes synaptic vesicles in the synaptic knob filled with acetylcholine to fuse with the membrane. Mitochondria, also known as the powerhouse for energy, are present in the synaptic knob. Acetylcholine is released from the synaptic knob by exocytosis into the synaptic gap. Acetylcholine is then picked up by receptors on the motor end plate of skeletal muscle. This action then stimulates the sodium-potassium channels to open, allowing the sodium ions to enter the muscle fiber and the potassium ions to exit. The increase in positive ions inside muscle cell initiates an action potential on the sarcolemma, or the cell membrane of the muscle cell. The action potential travels down the sarcolemma into the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calquestrin into the sarcoplasm of the muscle cell.
The high-energy myosin head leaps to bind on the exposed active sites on the thin filaments, forming a cross bridge. ATP molecule produced by the mitochondria in the muscle cell bind to the myosin head. ATP are then hydrolyzed to ADP plus phosphate. ADP remains for the moment on the myosin head. The energy released by this process causes the myosin head to swivel. This causes the thin filament to be pulled along the myosin, otherwise known as the power stroke. Calcium then floods over the thin filaments and binds to the troponin, pulling off the tropomyosin off the active site on the actin. In the process, the ADP plus phosphate is released and the head flexes to a bent, lower energy position. The next new ATP causes the myosin head to be released from the actin, thus allowing the head to be repositioned for another power stroke. Therefore, the myosin head will become energized as ATP is converted to ADP.
Contraction is then turned off by the following sequence of events. Calcium is pumped back into the sarcoplasmic reticulum and attached to calquestrin.
Allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle.
Acetylcholine at the neuromuscular junction is broken down by acetylcholine esterase, and this terminates the stream of action potentials along the muscle fiber surface.
This is the Muscle Contraction Project by
Courtney Splett,
John Kim,
Tess Robinson, not photoed, and Stephanie.